Chapter 7, Section 8, the Lac Operon. And here what we're going to be talking about over this section, the next couple of sections, are what are referred to as global control systems. Okay? This is a system that we've touched on in previous sections in this chapter that regulate a collection of genes, either as an operon or as a regulon. Okay? There's going to be activators, there's going to be repressors, signal molecules involved, some form of a two-component system. They'll activate, deactivate, and so on. When we go and we look at these global control systems in just E. coli, we see that there are systems for aerobic, anaerobic respiration, catabolite repression, heat shock, nitrogen utilization, oxidative stress, SOS response, and then just general response. The genes that are regulated can be anywhere from a dozen to hundreds. And it's all because of high, low, to how much cyclic AMP was made due to response of something. Is ammonia present, not present, was DNA damaged, and whatnot. The first one we're going to talk about, though, is the lactose operon. Okay, we've touched on the lactose operon in a previous section for this chapter. We talked about the Lac Operon as well as the maltose regulon. Remember the difference between operon and regulon? Operon is a collection of genes all together. The regulon is, well, you take an operon, you break it up into pieces, and you spread it around the chromosome. There. Here we're looking at what's called catabolite repression. Something, an intermediate product or the actual substrate or different substrate or a different molecule will repress or inactivate the use of this operon. Here, the Lac Operon is inherently repressed because all these cells are set up to always use any glucose present first. Doesn't matter if lactose is there, glucose comes first. Use up all the glucose, then go and look to see what other sugars are present. We see that if glucose is present, none of the enzymes for lactose or maltose usage will be transcribed, translated, so they are not even present in the cytoplasm. Glucose is gone, then you go to lactose. If both glucose and lactose are present at the same concentration at the same time, because of this repression mechanism, you see what is referred to as the glucose effect. Use up all glucose first, then go to any secondary sugars. Because of this, you can see what's referred to as a dioxic growth. So instead of that lag exponential stationary decline, you'll see lag exponential slight pause, exponential again, and then a true stationary. As the cells, the E. coli bacterial cells, switch from preferential sugar to secondary sugar, secondary carbon sources. So you see this nice sharp growth on glucose, and then this lag here as all the glucose is used up. So this plateau as new genes are being expressed, and it takes time. It's not going to happen in just minutes. It's going to take some time. And then you see another as it takes off due to the usage of the available lactose. This also corresponds when you looked at beta-galactosidase activity. How much is there? What's the activity levels? Beta-galactosidase is the enzyme that takes lactose and prepares it for entry into glycolysis. You can see here, as long as glucose is present and everything's set up for glycolysis of glucose, no beta-galactosidase present. Then once glucose is exhausted, you start to see this takes off. Quickly followed a few minutes later by new growth due to lactose now being used as the carbon energy source. This catabolite repression is going to be utilizing the protein known as CRP, cyclic AMP receptor protein. Okay, CRP is a cytoplasmic protein. It's not going to be embedded in the membrane. It's an activator protein. Okay, it's going to bind and activate transcription. Thing is, what binds to it? Cyclic AMP. Cyclic AMP, cyclic adenosine monophosphate. Its levels are going to be going up due to certain metabolic activities. So this is acting as a regulatory nucleotide. AMP levels high, binds CRP. So it's the normal ATP, adenosine 1,2,3 alpha, beta, gamma phosphate. Adenylyl cyclase cleaves off the terminal 2, phosphate 2, phosphate 3, beta, gamma, and does this cyclic back around, and here you get cyclic AMP. Okay, so our cyclic AMP and our CRP protein are going to come into play when it's dealing with yes or no whether or not the Lac Operon can be transcribed. Will the Lac genes be expressed? Turns out cyclic AMP levels have to be high. High enough that it's binding CRP protein, which then is binding to the control element of the Lac Operon. Here's the thing though, cyclic AMP levels are going to, well, adenylyl cyclase, I should say, is inhibited by high glucose levels. As glucose levels drop, cyclic AMP, the adenylyl cyclase activity can increase due to other signals in the cell. That means as glucose levels drop, adenylyl cyclase activity goes up. Also going up is the concentration of cyclic AMP. It gets high enough because glucose levels are low enough, binds the CRP, binds the control element, kicks off transcription. But just because cyclic AMP binds CRP doesn't mean that the Lac Operon can automatically be expressed, okay? Because there has to be the presence of lactose. Lactose has to be present and bind a repressor protein known as Lac-L. Because the repressor, remember the repressor here is automatically, once it's made, is automatically going to be bound to one of the elements of, the control element, sorry, of the Lac Operon acting as a roadblock. So let's look at this, in this illustration. Lac-L, which is separate from the Lac Operon, it's off on its own. It gets basal level expression no matter what. Lac-L forms a protein that's going to bind here at the operator sequence, which is upstream of the Lac Operon. No matter what, if RNA polymerase binds here at the promoter, Lac-L is there and it is acting as a roadblock, stopping all expression of the Lac Operon. To aid RNA polymerase in wanting to bind there has to be high enough levels of cyclic AMP. Okay. Remember, I just got done saying in the last slide, high levels of glucose inhibits adenylyl cyclase activity. Glucose levels drop, adenylyl cyclase activity goes up. Adenylyl cyclase activity goes up, so does cyclic AMP levels. Cyclic AMP, CRP protein. They bind the control element. They recruit an RNA polymerase. But it doesn't matter. LAC-L is still sitting there at the operator, hindering everything. Unless an inducer comes along. This inducer here for the Lac Operon happens to be lactose. So the presence of lactose in the cytoplasm, which means it has to be in the extracellular environment. Lactose binds a LAC-L, causing it to fall off. High levels of cyclic AMP due to the glucose being depleted or down far enough that cyclic AMP levels are up. Means that polymerase is bound here. And now polymerase can go through and express all the genes for lactose catabolism, the uptake and then the preparation of lactose to enter into the glycosidic pathway. No lactose present, doesn't matter if cyclic AMP levels are up, LAC-L will stay stuck there and no Lac Operon expression will occur. Switch over to maltose, switch one over to one of the other sugars and its corresponding operon.